Recording. Good morning, grade 9, my advisory. We are now in the continuation of our topic. We are still in the developments in genetics. Okay. Other developments in genetics. Again, last meeting, we have a film viewing of the creation, how to create a human through cloning. Okay. We also viewed on the screen the process, okay, on how to get a stem cell. Okay. Until now, if you're going to ask me, there are a lot of controversies with regards to and issues with regards to cloning and regards to stem cell therapy. Okay. If you're going to observe the video, it is actually talking about repairing, cell repairing through stem cell. Okay. Cloning. Okay. They need to clone an individual, okay, and get the stem cell out of it and insert it back to the original organism. So it's quite difficult because we are actually killing that's life already. So those are the issues in the other side of genetics, the developmental side of genetics. Okay, let's move. Through the advancement in the modern science, scientific research in genetics has become increasingly significant. Okay. Apparently, we cannot actually observe this in the Philippines simply because if you're going to ask me also, we don't have the technology needed. We don't have also the support needed in order to perform these experiments. This is still in the experimental procedures. Some of the developments in genetics are listed below. Okay, we have some of the developments in genetics. Let's have first mutation. Okay, mutation is a process that can be compared with selective breeding. Okay, it is done to produce new kinds of organisms and confine the characteristics that already exist in the population. Okay, an example of producing oil, digesting bacteria that can be used in cleaning up spill oils. Are you familiar with this? They tend to use bacteria. They actually created a bacteria in the laboratory that can digest oil. So meaning to say, if there are some spillage of oil in the ocean or in the seas, you will just release this bacteria in the water and it will clean up the water. Question there is, what if this bacteria will be ingested by us? That is always the case. What could be the effect of this in humanity? So that's always the question, always in the humanity. Okay, like for example, if we have this GMO, because this is actually GMO, part of genetically modified organism, we actually modify that organism in order to benefit us. Okay, this is GMO. What if we can actually digest this GMO or ingest this GMO? What will happen to us humans? What will happen to humanity? So that's always the case. Okay, let's move forward. Restriction enzymes are the proteins containing specific DNA sequences that are spliced from a gene. Okay, they are used to surround the DNA molecule, okay, at specific sites. They cut one strand of DNA into double helix at one point and the second strand at a different point. Okay, this is common. No, the restriction enzymes are actually used to restrict a particular gene. Okay, this is really common in genetic engineering. Okay, DNA insertion. Okay, DNA insertion is made when the DNA fragments containing the desired gene are obtained and inserted into the DNA that has been removed from the recipient cell. So the DNA insertion also is common in the genetic engineering. Okay, we cut the DNA of that particular same species and insert it back or insert it to another the same species, but different variety. Okay. Modern DNA sequencing, okay, has been used in the large-scale sequencing of human genome. Yeah, remember the Human Genome Project? They're actually using the human or the modern DNA sequencing. And has generated the complete sequence of many animals, plants, and microbial genomes. What is the benefit of having this or having the modern DNA sequencing technology? What do you think is the reason? Why do we need to identify, okay, all the DNA sequence, if possible all, but as of the moment, it is not yet complete. That's how um that's how large, or that's how huge the organisms living in this planet. Until now, it is still not complete. Even in the Amazon, there are a lot of organisms unidentified as of the moment. Amazon rainforest. Okay, the question there is, okay, you can always answer this one. What is the reason why do we need to identify all, if not all, most of the DNA sequence in all the organisms? Why do we need to do so? Remember that when you identify these DNA sequences, it takes money. Number one, it takes time. Number two, now for how many years we'll be dealing with those? What is the purpose of identifying this DNA sequencing and put it in a database? Why? Any idea, everybody? Any idea? Can you hear me?
Yes.
Yes.
All right. Why do you need to identify this DNA sequence or sequences? What do you think is the reason? You have your DNA sequence, correct? Other animals also have their own DNA sequence also. The question there is why? Do you have any idea?
No.
Right. Just to give you an idea, why do we need to identify these DNA sequences? In order for us, number one, to preserve an organism. Are you not aware that scientists until now are really eager, okay, to regenerate, okay, to regenerate dinosaurs? Have you heard about that? Because if the technology during the dinosaur period, Paleolithic period, has if we have the technology already there during that time, we can always preserve. Now, we have the technology now in our hands, okay, that can help humanity in preserving organisms. So, we need to what? Identify, again, we need to identify these DNA sequences in order for us to preserve a particular species from extinction. Okay? But then again, if you're going to ask me about preserving it through the DNA, we can always preserve the DNA. But the organism, I don't know. Okay? We cannot preserve. Until now, a lot of studies are actually into cryotherapy. Okay, cryopreserving. What do you mean by cryoture? Have you heard about freezing an organism and defreezing them after how many years? That's cryopreservation. Okay? Cryopreservation. That's to preserve an organism. But then again, that's a big question. Still, no, we cannot prolong, or we cannot actually hold them for a longer period of time. Simply because there is a cell degeneration. It will just degenerate. That's a big question. But we can always preserve the genes of that particular species. And hopefully in the future generations, we can actually make that certain organism. That's actually what the scientist is thinking about. Okay. That is the reason, the first reason why we need to identify the DNA. What else? The second main reason is for what? What do you think is the second main reason? Diba? If we are thinking about, diba, we're discussing about DNA insertion, and this is for the humanity. We're also discussing about the GMO, and this is still for the humanity. The modern DNA sequencing is still for the humanity. Okay. Why do we need to identify the DNA sequence? In order for us to remove the defective genes. Like for example, if a particular organism has hemophilia. Okay, hemophilia is the case wherein the bleeding, you cannot actually control the bleeding because of lack of certain clotting mechanism on the body. So it is actually genetic. We identified these genes as genetics. It is in the genes, in the line. So to prevent this one in the future generation, to prevent this one in the future offspring, we tend to remove, okay, the genes for the hemophilia and replace it with good genes. But of course, we need to identify first the particular DNA sequence in that certain gene. That's the second reason. This is for the benefit of humanity. Okay, for the betterment of humanity. That's the second reason. So again, the first reason why do we need to identify the DNA sequence or the DNA sequences is that number one, this is to preserve an organism. Okay, preserve in the sense that their DNA sequence is being preserved, and hopefully by future we can regenerate them. Okay, like dinosaurs. Okay. Number two is for the benefit of humanity, for the betterment of humanity. We create superhumans out of it, okay, by removing the defective genes and replacing it with the good genes, right? DNA sequencing is the arrangement of DNA-based sequence in a linear form. Linear means in line, okay? The knowledge of DNA sequence of genes has become indispensable in studying biological processes as well as the diagnostic and forensic studies. Okay. DNA fingerprinting. Okay. DNA fingerprinting is used in criminal investigations. Okay. This process is used for any specimen with DNA samples like blood, sperm, saliva. Like in the rape case, we usually use the sperm. Okay. If there are some sperm there, we need to check on the sperm. That's fingerprinting. The blood samples, the saliva, it contains also the DNA of a particular person or organism, and hair strands. Okay, if you're going to watch some international movies pertaining to investigations, they usually use hair. Okay, hair specimen, blood specimen, or even saliva. Okay, it matches the suspect's DNA with the DNA contained in the material or specimen left at the crime scene. I think we did also use this one in the Philippines, but only few. Remember that we are far behind with the technology, and hopefully also we can engage ourselves. We can actually embrace this technology because this is really great. Now, if you're going to ask me, uh, I really love to have this in the public. Okay, like we can always perform this in the public, but then again, because of the 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 cost of this is really expensive. Okay, again, DNA fingerprinting, we can actually check on, like for example, this is in the crime scene, the DNA sequence in the crime scene. Okay, and these are the DNA or the sequence that are actually found in the different suspects, because we have suspects, man. So we need to get their DNA or complete set of DNAs. Okay. So in the crime scene, we need to match okay, the DNA sequence. If somebody matches the correct DNA sequence, therefore, that person is the suspect. In that sense, we can most likely identify, okay, the correct or the person who did the particular crime. Okay, genetic engineering, as we always thought, okay, we already discussed this one. Genetic engineering is the process of manipulating genes to biotech or biotechnology. An organism produced through this process is called the GMO. Okay, genetically modified organism. Sure, what are those GMO? Okay, one good example of a GMO is your French fries in McDo and Jollibee. These are GMO. What else? Some of the rice that we have now is GMO. It can sustain drought. So what else? Corn. Okay, there are a lot of GMO which is actually generated in the corn. How? Well, definitely they had a full-grown corn, and talking about this, it actually what rejects pests. Okay, we still have around 3 minutes. I think we discussed a lot. I will be focusing by next meeting the discussion on the genetic engineering pertaining to genetically modified organisms and the effects of this in humanity. Right? Do you have any questions so far? Questions? Everybody, can you hear me?
Yes.
No,
Or
sir.
do you have any questions?
Yeah.
Right. I think that will
I
be all for
don't think
today's
so.
class. Okay, I think that will be all for today. And hopefully we can focus by next meeting on genetically modified organism under genetic engineering. Have a nice day, grade 9. Take care. Bye-bye.